Okay, I think we can just go ahead and get started here. So welcome. This is the single-cell RNA-seq analysis primary session 1. My name is Tyler Shapey. I'm a staff biostatistician in the Duke Bird Core. For those of you who don't know what that is, it's a core of statisticians, collaborative statisticians that work in the biostat department, and we collaborate across various researchers within the School of Medicine. Happy to talk more if you're interested in any sort of help we can give you from the Bird Core perspective. And if you're just joining us, I mentioned earlier, if you want to please, if you want to follow along, I think that would be beneficial for everybody. I have a lot of some code that we can follow along with. So we can start an on-demand session on the Duke Compute Cluster using these instructions. I think you've all probably done this before. And like I mentioned, you'll have to pull the latest content, and I'll actually show how to do that here in a second. So if you want to please launch an on-demand session, and we can get started shortly. Okay, so I'm going to go ahead and switch over to my browser here. So this is the on-demand homepage. If you click on this RStudio Workshop, CHSI RStudio Workshop, I think 4 hours should be fine. We want 4 CPUs and 32 memory. Everything else should be fine. Oh, I didn't mention, just use the R25 HIV/AIDS singularity image. Go ahead and launch. Then once this RStudio server button pops up, go ahead and click it, and that should launch an RStudio session. Okay. So the first thing you want to do is switch back to the project, the 2023-2024 HIV workshop project that will bring up the Git functionality within RStudio. And again, if this is your first time, maybe put something in the chat if you're on Zoom. Is it either of your first time attending? Okay, great. So you should have this project, I assume, already ready to pull up quickly. And we will need to pull the latest content. So if you'll go to this Git tab on the upper right here. Oh, and I guess I like dark mode, or like this dark mode. If anybody has an issue reading anything, please let me know, and I can switch. Sort of my preferred theme. Okay, so you'll probably, yours will look different because I've obviously been making changes here, but you should be able to use this blue down arrow to pull the latest changes. Don't click the tiny arrow next to it where it says pull with rebase. If you just do a regular pull, it should do something like this. And I believe there are instructions in case, instructions that Josh had written. I think if it's your first time pulling, you may get an error, or not an error, I guess a warning message saying that you can't pull, you have to do something first. So if you have an error message when you try to pull, try following the instructions in the main GitLab repo. And we can pause here and let everybody catch up, and I'll just check in before proceeding, make sure everybody has it pulled up. Were you both able to sort of pull the latest changes? Yep, okay, great. Does anybody on Zoom, is anybody having issues pulling the latest changes? If you want to speak up or put it in the chat, we can wait for you. Okay. Okay, all right, I think we are good to go. So just a quick note, if you click this little R logo in the file browser over here, it should bring you to the home location for this project, which is the main page of the GitLab repo. We're gonna be working in this assays directory here, and there should be a sc_RNA-Seq directory within that. And within that directory, there is a notebooks directory. And within that directory, there is a file 03 day 5 single cell session 1 R markdown. That's what we'll be working with today. So if you want to just go ahead and click that to open it, you should see a R markdown notebook. I'll be working in the visual view here for the presentation, assuming it loads. It's just being slow. Okay, all right. Well, at any rate, we can get started. So just a couple of intro points. Again, my name is Tyler Shapey. I'm a statistician in the Duke Corps. I did want to just sort of ask quickly before we begin, does anybody have prior single cell analysis experience? Do either of you have sort of any experience in the past? Very little, okay.
The last 2 weeks.
2 weeks, okay. All right, great. Anybody on Zoom have any experience prior this course? Okay, all right, great. Okay, always good to sort of know where people are starting from, and we can kind of cater to that. Okay, so I guess I did want to mention that please ask questions as they come up. I kind of want this to be more of an informal sort of workshop, not so much like a formal presentation. I think there'll be plenty of questions as we go along. I do also have some exercises built into the code where we're going to take a break and do some interactive exercises, and I can kind of help with anything with that. So we'll keep it light. For the format of the workshop, I'll be kind of alternating between slides, which I'm showing here, and the RStudio session. Like I mentioned, we'll be walking through that R markdown file. A couple of disclaimers. We'll get into this more, but I'm only going to show sort of a sample of tools and methods. There are literally thousands of methods out there for single cell analyses. I'll cover that in a little bit, but just to note that this is not like an exhaustive covering of all the methods, and I think there are kind of choices that I've made here in presenting what I'm showing. So just keeping that in mind. And then this guide is going to be sort of a practical, guide from a practitioner's perspective. I'm an applied statistician, so I won't be kind of going into a lot of theory, or we'll be covering some of the methods in depth, but not to maybe the extent that you would in another more focused session or setting. Okay, so with that, we can go ahead and sort of get started here. The first thing I just wanted to show is that there are many tools to do single cell analysis. This kind of table here is a summary of just ecosystems, common ecosystems, I would say, within R and Python languages, just to show that Seurat is one among many. Seurat is what we'll be using today. It's one among many possible sort of toolkits, let's call them, that you can use. Bioconductor has their own sort of set of tools. Monocle is sort of like its own Seurat-like thing, except it's like a competitor. And then within Python, I think the most commonly used suite of tools is scNPy. All of these ecosystems have kind of a lot of overlapping functionality, albeit with different methods involved. They each have their own data object. You can do QC, quality checking, normalization, batch correction. There are also many tools that are able to be run in either or multiple ecosystems. So for example, you can run Harmony integration in Seurat or scNPy. And so we'll cover some of that, but just to kind of give you an idea of the breadth of what's out there. On the right, this table is just a table that I pulled from a paper that was comparing trajectory analysis methods. You can clearly see there's a lot of different methods and sort of subtypes of methods. I don't expect you to be able to kind of read all that, but just to give you an idea of how many there are. And then on the bottom is a similar table from a paper that was comparing sample integration methods across R and Python tools. So there's a lot out there, and it's complicated. And it's honestly pretty hard to sort of follow sometimes. So I actually found this website, which I didn't know about until a week or two ago. scRNA-Tools.org is a website devoted entirely just to tracking the number of tools that are out there for single cell analyses. You can see, well, maybe you can't see, but as of 2024, there were nearly 1,800 cataloged tools in this database of tools. And so, you know, it's overwhelming, the number of things that are out there. You can see most of them are either R or Python, kind of a minority in C++ or MATLAB. On the bottom, you can see a lot of the tools are visualization, dimension reduction, clustering, sample integration, are sort of the top categories there. So I think sometimes it's challenging to navigate all these tools in this ecosystem. Okay, so I also wanted to mention that for single cell analyses, there can be many different goals starting out. And so I think part of the trick is sort of finding a path and a set of tools that accomplish your goal. So I kind of have it broken down here by condition, single versus multiple conditions. Within a single condition, so that would be, for example, like you have one type of subject or one organ or, you know, something along those lines. You might be interested in identifying cell types within that tissue or condition. You might be interested in describing or discovering novel or rare cell types. I'm part of a collaboration that's trying to discover and describe senescent cells from a catalog of patients from different organs. So that's sort of the goal there. And you could also maybe be interested in characterizing developmental trajectories of cells. So then you get over to this multiple condition category where you might have more of a sort of traditional setup for an experiment. You might have two different tissue types. You might have two different conditions. You might have a treatment group and a control group. And you may be interested in measuring changes in gene expression or changes in cell type abundances, and quite a few other potential questions. And so I think when you talk about single cell, you have to kind of understand that there are a lot of facets to it and a lot of tools that you can use. So it's good to sort of have an idea before you start where you might want to go and what tools will get you there. I'll also just briefly mention that there's a sort of newer concept of multimodal data. So when people talk about modalities, they talk about different kind of, I guess I would say, ways or methods of measuring things for the same cells. So this top row is, you know, gene expression by cell. That's single cell RNA-seq. That's what we're going to be talking about today. There are other modalities, chromatin accessibility. You're looking at epigenetic, epigenetic patterns. Let's say that's single cell attack-seq. Cell ranger attack can do that. Cell surface proteins, if you're interested in characterizing those, there's a specific assay you can do called site-seq. And then if you're interested in immune receptors, you can also sequence those with, you know, T cell or B cell receptor sequencing. And these can often, well, they can be done on the same exact cells. So you can do basically multimodal assays of the literal same cells and measure multiple things on them. So pretty cool that you can do that. We won't be covering them, but it's just something to keep in mind as you're designing an experiment or talking to an investigator. That's a possibility. Grayed out here in the bottom is spatial gene expression. That's actually not by cell, at least not yet that I'm aware of, but that can also be done. Basically, people are interested in expression by location within a tissue. Okay. So along with all these tools and modalities and questions are many challenges. It's a challenging type of data to work with, and a lot of the tools that have been developed are basically have been developed to deal with these multiple challenges. So this is sort of just a summary of the most common ones and some of the biggest ones. We'll basically be walking through each of these rows in the table throughout the workshop, these next two workshops. And we'll see how a lot of the pre-processing steps basically address each of these challenges or attempts to address each of these challenges in turn. And so I guess maybe just to quickly cover them, there's PCR amplification bias, there are empty droplets that occur during library preparation, there are doublets that occur, you can have poor cell quality among your pool of cells, there's sequencing variation by cell, there are these confounding gene expression patterns from cell cycles and other things, and then there's sort of leftover sample level batch corrections that people want to account for. And then finally, there's this sort of ever-present dropout phenomenon, which is kind of like an ongoing debate about how best to deal with it. So we will kind of go over each of these. I did want to mention that given everything I've shown so far, an analysis is sort of like a single pathway through sort of this confusing tree of possibilities that you can do, right? You can, at each of these bifurcations, you maybe can go multiple different directions, depending on the tool you use, or the choice of parameter you might use, or the metric you use to filter things. And so I think it's best to sort of consider that, and people have, publish on kind of trying different things and picking one path through this forest of methods. Okay, so with that, I did want to cover these three highlighted challenges before we actually kind of begin the interactive workshop part. So I just wanted to cover, I think Josh had covered UMIs, at least he told me he covered UMIs to some extent, but I'll kind of go over those a little bit more. And then this empty droplets problem, I'll talk about the empty drops algorithm that's been developed. And then finally, I'll touch on dropout a little bit. Are there any questions before we move on? Questions on Zoom? Okay. Okay, so the first challenge that I wanted to mention is this thing called PCR amplification bias. And the table kind of mentioned UMIs, which are unique molecular identifiers, as a solution for this challenge. So I'll just really quickly cover, hopefully quickly cover what UMIs do and why people use them and how they sort of overcome this challenge. And so this figure from Islam et al. 2014 basically shows this kind of nice illustration here with two cells. Cell one has these transcripts within them, within it, messenger RNA molecules. Cell two has the same. If we imagine that the colors correspond to a specific transcript, then we can see that some of the colors are shared across the cells. So maybe the cells are expressing similar genes or the same genes. If we focus on the blue transcripts, can you guys see the, yeah, you can, okay. The blue transcripts, so cell one has 4 blue transcripts, cell 2 has 2. So let's just imagine that's, some gene as it's being expressed in both cells. Cell 1 has double, the expression of cell 2. So what happens in the library preparation stages, there's reverse transcription to transcribe that messenger RNA into cDNA. In that same step, the molecules are barcoded, and in kind of modern methods, they're also labeled with unique molecular identifiers. So essentially what happens is each of these little strands gets its own unique tag indicated by the colors here in the bottom. So you can see each of the 4 blue molecules in cell 1 has its unique color. At this point, I would point out that some messenger RNAs do not get tagged with a UMI, and so are thus lost basically to the analysis at this step in the analysis. And I'll kind of circle back to that later. So this next part of the illustration shows that after PCR amplification, so that's where these cDNA molecules are, they go through PCR, they're copied essentially during that process. And you can see now that some of the, like all of the molecules with the green tag have now been copied. There's multiple copies of them in both cells, cell 1 and cell 2. So then after amplification occurs, we sequence the products of that amplification, and we basically just count how many transcripts we observe, how many reads. So there's sort of 2 different ways of counting. Ultimately, sort of the raw data, right? You're counting the actual sequencing reads. That corresponds to each molecule from the post-amplification step. And so in cell 1, we can see that that blue molecule where we started off with 4 now has 15 copies that were all sequenced. So those show up as 15 different reads. I guess read pairs to be exact. And for cell 2, the original 2 transcripts now are represented by 8 reads. So you can see that part of the process, right, the PCR amplification was done because we needed more input to the actual sequencing process. So we needed to basically increase our DNA concentration. But the byproduct of that is sort of this, amplification or multiplication of the molecules. The problem is that PCR is not sort of a perfect reaction. It's not uniform across all of the original molecules. There can be biases. There's also randomness involved. So like if, one gene is, if a gene has a very few number of transcripts to start in the amplification process, it might ultimately not be amplified kind of proportionately compared to other genes. And so you can have this PCR bias, and you can also have this kind of randomness that's added to the process. So the UMI is basically what you do is you take your reads, and using that tag, that identifier that you ligated to the molecule, you can collapse those reads back down to the original UMI, the original molecule count, basically. So that sort of pretty much undoes that kind of noise addition that's created in the PCR step. Okay, moving on. There's this other challenge of empty droplets. So this occurs during the phase where the gems or gel in bead emulsions are created. So again, I think Josh covered this, but basically there's this microfluidics device during library preparation where these beads, which contain the molecular identifiers and also the oligos, are flowed through this device, and cells are also flowed at this juncture. Basically, the aqueous solution then goes into this oil, which forms these droplets. And the device is tuned so that there's sort of this balance between having a ton of empty droplets and having multiplets. So I think they've sort of tuned it to sort of be right in the middle or to optimize that. But what you end up with are by design, a lot of empty droplets, which are pictured in this diagram. You can see gel bead A, gel bead C, and gel bead D, all are droplets without any cells in them. And so the problem with this is that there can be sort of ambient RNA. So the cells that are coming in through this channel here, it's been found that there can be ambient messenger RNA. And so when the empty bead, there's no cell, but it actually can encapsulate some background RNA. And of course, if that RNA is present, it will be reverse transcribed, it will be amplified, and you'll end up with some amount of reads and some amount of UMI counts for that bead in the downstream analyses. So really the question is, how do we distinguish empty droplets that don't have any cells from droplets that truly do have cells? You'll also notice that there is one bead here that has 2 cells. So we'll cover that later. Those are called doublets or multiplets, and we'll think about that later. But so one of the answers, or one of the methods, I guess, for accounting for this or dealing with this empty droplet problem is the empty drops algorithm. It was published by Lunn et al. In 2019. And it was very successful to the extent where it was actually integrated into the Cell Ranger pipeline automatically. So I'll kind of cover sort of at a high level what empty drops is doing. And we can see how it's trying to determine what is a true droplet, or what is a true droplet with a cell in it. So the first step is to identify cells that have high RNA content. So the way that the algorithm does this is it applies a cutoff of total UMI counts per barcode or per droplet, you can think of it. So I put this figure is from 10x on the right. I put this horizontal orange line here to indicate that threshold or cutoff value that would have been used for this particular run. That threshold is sort of determined either by it's determined by the expected number of cells, and that value is either fixed, which was the case in earlier Cell Ranger versions, or it's actually now estimated in more recent Cell Ranger versions. by sample. And I won't actually go over how that's done. It's kind of a rather complicated process, but suffice it to say that there's a threshold value that you say every droplet above, UMI counts, let's say 2,000 in this case. If a droplet has more than 2,000 total UMIs, we're going to call it a cell. So you can see, sorry, I probably should have explained this figure more on the bottom, but it's sort of a plot of if you order the barcodes by UMIs, and then you can make a plot like this, basically where, and of course it's on the log scale, log 10 scale here, but you can see that roughly about maybe 300 or 400 cells or barcodes have UMIs greater than 2,000 in this case, and the rest are considered background. They don't meet that threshold. The second step in the algorithm is called refinement. And so the next step is basically you do sort of the opposite. You identify almost surely empty droplets with a very low UMI count. So on the figure, it would be a line, something like this. I just made this line up, but you can imagine saying any droplet with fewer than 5 total UMIs is going to be, we're going to call that an empty droplet for the purposes of finding this. So with those, it looks like probably you'd have thousands likely of droplets meeting that criteria, which would be all the droplets below that blue line. And what you do with those droplets is you actually model the gene expression of them. So you'll come up with, it's actually like a negative binomial model where it's modeling basically each gene that's found in the empty droplets, and it's modeling the expression of those genes. And then you compare the possibly empty droplets against that model. So the possibly empty droplets are those that are in between these two lines on the graph, right? We didn't call them for sure cells, and we didn't call them for sure empty droplets. They're sort of in the gray area. So we take all of those droplets and we actually compare their gene expression, their observed gene expression, against this model that we fit using the empty droplets. And there's details in there that I'm skipping over, but essentially if the barcodes deviate significantly from the model for the empty droplets, then they're called a true cell containing droplet. And if they don't deviate significantly, then they're called background. So this is the other half of the figure presented by 10x, showing sort of how empty drops can improve on the cell calling during this phase. So the previous method with just a UMI cutoff, identified something like maybe 300 droplets with cells. And this with empty drops, this new method, you can confidently call maybe even 2,000 cells. So you really do increase the number of cells that you can assay with this by sort of having this improved way of finding empty droplets. Any questions so far? Okay, so moving on to this third challenge that I wanted to cover before we kind of get going. This challenge is called dropout. And so what this is basically the phenomenon where you see a gene is expressed but not detected through sequencing. So it's sort of a hard thing to say for sure that is happening, right? You can't know for sure that a gene is being expressed. But people have observed that there are, in many cases, in many studies, cells where there are genes not expressed at all, but you would expect them to be expressed. Things like housekeeping genes or other common genes that you actually just don't see any counts for. And so this is actually an ongoing discussion in the literature. This figure that I have here is from a paper from 2022, where basically they were responding to a debate about how to best model this dropout phenomenon. And essentially what they created here is each of the steps of the library preparation process and how each of those steps can basically contribute zeros to the data set, the ultimate count matrix that you get at the end. And you can think of each of these steps basically as like a sampling of the previous pool of cells from the previous step or molecules. So we can kind of walk through the rows of this. On the left, kind of the left half of this is a little bit less relevant. It's kind of just depicting the biology of what's happening. But in this B panel, I think is a little bit more relevant for our purposes here. So across the columns are different genes. And they set it up so that each of these genes actually either gets dropped out or not by a separate mechanism in the process. So the first row of the table is showing, it says RNA polymerase binding, but basically what they're trying to show is that, some genes are simply not expressed, right? In a cell, it can just not be expressing that gene. That would be a biological zero, right? It's truly a zero count because it's not actually expressing that cell at the time of sample preparation. Gene 2 over here is being excluded from the second row, and it says messenger RNA existing in the cell. What they mean by that is that sometimes there can be degradation of the messenger RNA kind of during the sample preparation phase. So it could be possible that a gene truly does have messenger RNA being produced and it's being expressed in the cell, but just through the processes of the kind of library prep, that messenger RNA is degraded and it gets removed from the library. So that's a biological zero they're talking about as well. It is a biological process where the RNA degraded. It was nothing sort of from our side of technical error. It is a true biological process. So beyond that, there is the step after you extract the messenger RNA, you synthesize cDNA with it. During that step, basically, it's not a perfect process. Not all of the messenger RNA molecules are indeed reverse transcribed into cDNA. So you can have these technical zeros where the messenger RNA was present, it was being expressed, but it's actually dropped at this phase of the pipeline. And then beyond that, the next step is this PCR amplification step. Kind of a similar story. It's not a perfect process, right? Not every cDNA molecule gets amplified to the same extent. If there are very few cDNA molecules to start with, they might not be amplified much at all, and you might end up with a zero. And so that's a sampling zero according to Zhang et al. 2022. And then finally, you end up sequencing that library, those libraries that you created after the PCR step. Not all of those molecules are actually successfully sequenced. And so basically, like every step, every row in this table is sort of the sampling procedure that happens. And so what that does is it leads to noise in the gene expression signal, right? There was a truth that we tried to capture, but at every step we're introducing sort of this noise. And that noise can basically show up as zero inflation in the gene matrix. And that is what people refer to as dropout. So our goal basically through these analyses is to distinguish biological signal from technical noise. And like I mentioned, it's an ongoing area of research how to sort of best model this noise. People have said, oh, you should use a zero inflated model. No, negative binomial is sufficient. So I think, there's seemingly no clear answer yet. What is the best method? So people have competing methods, and we'll kind of cover it. Okay, so I see in the chat, sorry, I joined late. Got me to the markdown. Let me, so the markdown is actually in, if you follow the instructions on the first link, you'll need to start a DCC session, and then you'll need to pull the latest changes from the GitLab repo using the pull feature, which I think is outlined in one of the articles in the on the GitLab. And then I guess I can just quickly show where the notebooks are. So from the home location, I think you all can see this. Yeah, from the home location, it should be in Assays, SC_RNA-Seq, and notebooks. And then it's day 5, single cell, session 1. Okay. Oh, shoot. Okay. Are there any questions about dropout that I can try to cover? Okay. Josh is working on the link. Okay. Okay, so returning to this many challenges table, I think what I have highlighted here are the challenges that we're going to cover in this sort of notebook we're going to work through. I mentioned UMIs and empty drops. Those are integrated into Cell Ranger. And the UMIs are, they were kind of a step that we took during library preparation. Obviously, we didn't do it. Whoever did that for the samples we're using. So those sort of are tools that are already implemented at this phase in our pipeline. So we'll focus on these downstream ones. Okay. All right, so I wanted to maybe just take a 5-minute break here really quick, and we can, afterward, we're gonna kind of go into the notebook portion. So let's maybe just take a little bit. And I can answer any questions if you guys have, and if you have anything in the chat or in the Zoom, please put it in the chat. Okay, so maybe we can resume here unless anybody has objections. All right. So before we dive into the notebook, I did just want to briefly cover the data that we'll be working with. This is a study from 2022. It was published in 2022 from Peel et al. They published this in Cell. I put a geo ID there for those of you who are interested in maybe looking it up. It's a publicly available data set. They ended up having 59 total samples or subjects. We're just going to look at 6 of those that I kind of just pulled. And the tissue that they extracted these cells from was cerebrospinal fluid. And their goal of the study was to examine immune cell changes in the CSF due to aging and or Alzheimer's disease. So they basically sampled from different patients that were of different ages, mostly of older ages, and they also had known people with Alzheimer's diagnosis. They used the 10x chromium 5' V2 chemistry library preparation. That 5' enabled them to also do single cell TCR sequencing. We won't use that, although it is available if you're interested. That would be kind of a nice data set actually to sort of practice a multi-ohm analysis if that's something that you might be doing in the future. And so yeah, here's sort of just a summary of the 6 samples that we have for today. You can see 3 healthy and 3 with Alzheimer's diagnosis. Roughly similar ages except for that one person, subject 4, and then a kind of heterogeneous number of cells, which is not atypical for an experiment such as this. So that'll be something we'll be keeping in mind, but anywhere between 337 and 2,600 cells, basically. Okay, so this is like an overall kind of overview, I guess, of the workflow that we'll be doing in the 2 sessions. The top row is what I hope to cover in this first session, although I think we might be, based on kind of me practicing this, I think we'll probably stop short of the normalization step for today and carry that on to next week's. So we have our cell ranger count output in the upper left. We'll be creating Seurat objects from that output, removing multiplets, filtering cells and genes, and then the normalization step. All of those steps will be done with the samples kind of kept separate, as is recommended. And then the lower half of the pipeline, PCA, dimension reduction, sample integration, cell clustering, UMAP, and annotation will be performed with the samples actually combined. And we'll cover the second half next week. Okay, so the first step that we'll tackle today is creating Seurat objects. And I want to kind of give a quick intro to the raw data and how it sort of translates into a Seurat object, and then a little bit about the Seurat object itself. So the raw data output from Cell Ranger comes as 3 files, and it's basically a kind of space-saving representation of a sparse matrix. So we know a lot of the count matrix, which is genes by cells, and the values in that matrix are UMI counts. That's going to be a very sparse matrix generally, and so it's not very efficient to sort of store that and use that in that format. So what Cell Ranger does is actually give you 3 files that you can reproduce that matrix with. So the first file is just called barcodes, and it's basically just a list of the barcodes for each cell. So, cell C1 through Cn, let's call it. It's just a list of the actual nucleotide sequence for the barcode. I guess I should mention all of these three files are TSV files, so they're just raw text files that you get. The genes file is a list of the genes that were observed in the sample, so across all the cells, which genes were observed, and their gene name, and as well a gene ID, which kind of is a universal lookup ensemble gene ID. And then finally, we have the matrix file, which is, you can think of it as sort of a matrix file, but in long format with the 0 values removed. And so every row in that matrix file is a gene and cell combination. So for example, in the first row here, we have gene 1 within cell 1. And the third column of that file is actually just the UMI counts. So gene 1 in cell 1 had 3 UMI counts recorded. Similarly, gene 4 in cell 1 had 1 UMI, and etc. So that's obviously a huge file. It's every gene and every cell combination that we observed. But omitted from that is the 0 counts. And so that's partly what makes it more efficient. Okay, so moving kind of to the Seurat object. Ultimately, Seurat does store a sparse matrix within it, and we'll see that kind of when we walk through. But that sparse matrix looks like this. The cells are columns, and the genes are rows in that sparse matrix. And within Seurat, there's actually a lot of other stuff, which I'll show in a second. But kind of the main bulk of the data is in these 5 bins, you can think of them. So the counts data is kind of the first thing. It's basically the raw counts, the raw sparse matrix that you get from Cell Ranger is stored in the counts slot. And I apologize, Seurat has actually gone through an update. They just released not that long ago version 5 of Seurat, and they've actually changed the nomenclature of the different kind of elements of the Seurat data. So I'll probably maybe go back and forth between names, but what used to be called slots is now called layers. So counts is actually a layer. Data is a layer. Scale data is a layer. So counts is the raw counts, like I mentioned, the raw UMIs. Data typically holds some transform version of the counts. So for example, log, if you log transform your counts, you would put that in the data layer. And then scale data is usually reserved for an additional scaling transformation. We're going to be using this scTransform function. So scTransform actually puts in these residual values, and we'll talk about that later. But scale data sort of holds these transformed data slots, and that's typically for what are identified as variable genes in the Seurat object. So it's a subset of the gene. And I guess I didn't mention, but sort of in this diagram on the right, cells are kind of across the columns and genes are on the rows. So these 2 remaining other layers or other elements are not data, strictly speaking, or transform data. So the top one is metadata. And I put it sideways because it's actually a data frame in the Seurat object. It's a data frame where the cells are rows, confusingly. So it's kind of like transposed a little bit from the counts matrix. So cells are rows in that metadata slot, and columns are just the metadata elements that you have. And then on the bottom, we have reductions, and reductions holds reduced dimensional data. So typically, and we'll see this later, typically you start with maybe 10 to 15,000 genes and the expression, and you might want to reduce that to a few, maybe a 3-dimensional space or a 2-dimensional space. So reductions can hold that reduced dimension space. It has the same number of rows as there are cells, but it just holds different information, a reduced space. Here's a full, I'm not going to cover everything, but this is actually what the full Seurat object, if you were to expand all of the elements of it, this is sort of what it looks like. The assays bucket on the top, or slot, holds, like I said, kind of the bulk of the data. You can see here they say assay 5. That's their new type of Seurat object, which is relatively new for me. I'm actually used to working with previous versions. So within that assay 5, there's layers, cells, and features. Within layers, you can see we have counts, data, and scale data. The metadata lives in a separate slot entirely, which actually means that it can apply to multiple assays. And it does apply to multiple assays, and it's actually a data frame. And I'll cover some of these other things. You can see reductions down here. Neighbors holds usually like a k-nearest neighbor network information. Images can hold spatial, if you have spatial transcriptomics, you can store images there. And there's a couple other miscellaneous things that I won't mention. Okay, so we can move on to code section 1 in the Markdown. So why don't we transition over there? And like I said, if you guys would feel free to kind of follow along, there will be some exercises that we'll try to run through, which would require you to sort of have run everything up until that point. So if you want to just follow along with me, that'd be probably the best way to do it. So we can go ahead and just run, load our libraries here. If this will run, it seems like it's being slow. I hope it's not going to be super slow. Still, yeah. There we go. Okay. Okay, so code section 1, we'll load the data and create Seurat objects. So I just wanted to first kind of look at the cell ranger output. I sort of described what it looks like, but I did want to kind of show that actually in the file. So the first thing we'll do is just examine the output directories so we can get a sense of what the actual output of cell ranger looks like. So I think you guys ran cell ranger for 1 sample with Josh. What I did was run cell ranger for all 6, and then I dumped it into a directory. That directory is cell ranger out prep dir, which is defined in this source file, in this config file. So if you ran this first chunk, you would have sourced that config, and this location is defined for you. So if you do list.files, we can just take a look at the 6 samples. Their names are sort of nonsensical, but SRR is like the database where I obtain the sequences from. These are just unique identifiers from that. And we can take a look within one of those directories. So we just pick one. You can see that, oops, I think the output is cut off here. One second. Man, this is being really slow. Well, there is an outs directory within that directory for the sample, and this code should show you the files that are in that outs directory, and it's currently not running. Okay, well, basically this outs directory contains the 3 files that I was mentioning earlier, so we can just move on. I did want to mention, I think you looked at this with Josh, but there is a helpful web summary HTML file that Cell Ranger creates when it runs. And I won't, for the purposes of time, I won't sort of open that up, but just to mention that you can navigate to that in RStudio and open it up and take a look at it, which you guys probably did previously. It does give a lot of good information. If you just want to quickly look at the kind of initial quality of the run, that's a really good place to start.
Raw feature BC matrix, then filtered feature BC matrix.
Okay, yeah, great. So yours is obviously running. There's a filtered feature. I don't know why this is not being cooperative. There's a filtered feature BC matrix folder in there, and that contains the files that we're going to be working with. The filtered refers to the fact that empty drops was run on that, and so that's already been the empty droplets have been removed from that. The raw feature is not empty drops in case you want to do your own, for example. Okay, so the raw data files, like I mentioned, there's 3 of them. I won't kind of rehash all of this, but we can kind of take a look at what each of them look like for one sample. So this is the barcodes file, contains the cell barcodes. So you can see I just sort of loaded the first 10 lines of this file. Again, it's just sort of it's a gzipped TSV file. Shalene, you just mentioned the filtered feature BC matrix. So that's what this file path is, pointing to that filtered feature BC matrix directory within one of the samples. And I'm just loading it with read TSV. So yeah, this is just the first 10 barcodes. You can see it's just literally the nucleotide sequences for the first 10 cells. Okay, so the features file is going to be in the same location. And again, it contains 1 row for each unique gene observed in the data set. And you can see the first column is actually an ensemble gene ID, which is useful for kind of looking up information about that gene later on. That should be a pretty unique identifier for the gene. And then sort of a common gene name is the second column, and the assay that the gene was found in. For our case, it's always going to be gene expression. The matrix file is kind of the most interesting, and again, it reports for every row, it's a combination of a gene and a cell. It does actually have a couple of metadata in the first few rows. The first column is the gene ID, which is given in the genes.tsv. The second column is the cell ID as the line number in the barcodes file, and then the third column is the UMI count for that cell gene combination. So we can kind of load that and take a look.
The first row has sequences total.
Yes, so that's kind of what I was mentioning. So the first 2 rows contain some like metadata, on the matrix, and then it's kind of got the software version for Seurat. And then yeah, this third row is actually the sums of all of the columns. So you can see we actually, since the second column is cells, we have 344 unique cells. The total UMI counts is going to be 817,000. And then there's 36,601 unique gene cell combinations. That's not genes. That's actually a gene by cell combinations, because the genes can be repeated, among cells for that. Yeah, that line is not very useful. I usually, actually, Seurat doesn't use it as far as I'm aware. Okay, so we can go ahead and load the data into Seurat and create a Seurat object. First, a couple of things to note. So like I mentioned, we ran Cell Ranger on each sample separately. So the workflow, we're going to actually keep each of the samples separate through this first part of the workflow. So we'll be creating separate Seurat objects for each one. And then we'll combine these later into one large Seurat object. And to do all of that, we're going to be using R's list object to store the Seurat objects into a list. It'll simplify a couple of things. And then, we're going to perform some basic sample-wise filtering when we create the Seurat object, and I'll cover that in a little bit. So I just kind of wanted to give a brief primer. I'm not aware of exactly what you guys covered in the previous R workshop sessions. I didn't go back and look at all of it, but I did just want to have everybody on the same page for lapply and lists. So I have a couple examples here of using lapply. And so basically, in a nutshell, lapply is a built-in base R function for looping over anything, and it always outputs a list. So it's very very useful in many ways. I think some people have kind of moved on to using purrr, which is a tidy package where you can actually use this function called map, and it does it in different syntax. But I think for our purposes, it's useful to use lapply. It's a little bit more verbose and a little bit maybe clearer what it's doing. So here is an example, a couple of examples of using lapply. So the goal is going to be to create a list of data frames with a data frame for each sample that contains the path to the corresponding Cell Ranger output. So this is just kind of an arbitrary goal that I came up with to use this lapply function. So the first thing we do is create a vector of sample names. And I did that by extracting the names of the directories in the CellRanger output directory. So listers just kind of gives you a list of the directory names in a directory of your choice. And I'm saying recursive false because I don't want the full path. And full names the recursive false, I don't want any subdirectories to be listed. And full names, I don't want the full path of that directory. So this is just going to print out basically those 6 sample names that we saw earlier. So this is what I'm going to use to loop over. This is a vector, of six sample names. So lapply, the syntax looks often like this. At least this is the way I usually use it. This is where you're kind of defining a function that you're applying on the fly. And I've found it useful to do it this way. So, we're storing all the output as example loop out. Then we call lapply and we say we're looping over some object. That object in this case is going to be that vector of 6 sample names. And then we can define a function, an on-the-fly function, and we can define any number of variables we want to. I tend to sort of use a variable name that makes sense. So, when we loop over these sample names, there's going to be a current sample that we're looping on. So I'm just going to call that current sample cur_sample. Okay, then we have this curly brace. So that's going to be the actual function that we're applying to each iteration of this loop. Within that function, we can do basically anything we want. In this case, what I'm doing is saving the output of this data frame as out. That out will only exist within the loop iteration, and then it will be deleted for each loop iteration. What we're going to create is a data frame with 2 columns. And so the first column is just going to be the sample name. So I'm just saying sample equals cur sample. And the second column is the filter matrix path. And I'm defining that by using this file path function. And we're just passing basically the CellRanger output dir. And the key here is that we're giving it the current sample name, because that's how the output is organized, is by sample name. And so this is going to create a path to the directory where, the output directory, the current sample name, and then we know that the outputs for that sample are within the outs folder within the filtered feature BC matrix folder. So that will basically create a full path to the filtered feature output from CellRanger. And then at the end of the loop, we just return the output data frame for the current sample. So if we go ahead and run this, it shouldn't take too long, and it'll give us output loop, example loop.out. And if you look in your environment, you can see that it's a list of 6. So we can go ahead and take a look at what it has. So this str function kind of gives you some basic information about an object in your environment. You can see that example.loop.out is a list of 6 things. They're all data frames with 1 observation and 2 columns, and they contain sample and filter matrix.path. We can also access or slice this list using double brackets. So if we do example.loop.out double bracket 1 is going to give us just the first element of that list. And you can see that it's a data frame with 1 observation and 2 columns. Sample is this first sample, and the full path is stored here. Okay, any questions about how lapply or looping works so far? Okay. All right, so I next wanted to show how we actually create a Seurat object for a single sample. So the goal is we're going to take the first sample and we're going to make 1 Seurat object. So the first step is actually creating a sparse matrix. And so we create that from the genes and barcodes files that we examined earlier, using the Seurat function read 10x. And it's a pretty straightforward process. You basically use read 10x, and you tell it the directory of the path to the directory. In our case, this filtered feature BC matrix. I'm giving the path using this file path function I probably should have maybe named this differently because it kind of looks like we're in a loop based on my previous syntax or naming. But cur sample, actually I defined way up here when I loaded this first one. Where is it? I thought I defined that. Well, anyways, this is one sample, basically, one sample name. And we are specifying that the column in the features TSV file is number 2 for the gene names. If you recall, the genes file had sort of the ensemble IDs in the first column and the gene names in the second column. And then for the cell file, we're specifying that column 1 in the barcodes file is used for the cell, it's the only column. So that's pretty obvious. So if we run this, we will create a sparse matrix object, and we can take a look at what it has. So you can see that's returned a object of type DGC matrix. This is sort of the standard, or I guess a standard method for storing a sparse matrix within R. It's a bit convoluted how this maps back to the original matrix. I do have some information here if you want to read further about what each of the kind of elements of this object are, but suffice it to say that you can basically exactly reproduce the matrix, the full matrix using this information. We also know that the features are stored as rows and the cells are stored as columns. Okay, so then kind of the next step is that we can create a Seurat object from this sparse matrix using create Seurat object function. And at the same time, we can also perform some basic filtering steps here. And so the first filter step is that we're going to say that the minimum number of genes, the minimum number of cells for a given gene has to be 3. And this is a pretty standard way of defining this filter. So we're not going to use, we're not going to include any genes that are seen in fewer than 3 cells. Any such gene would be extremely rare and not very informative for downstream analyses. It also does imply that we may not be able to find subpopulations of cells that consist of less than 3 cells. So something to keep in mind, if you are looking for extremely rare cell types, this might not be something you want to do, but it's a pretty standard way of filtering at this step. And then similarly, we're going to say that we need at least 200 genes for a cell, with the logic that cells with fewer genes are likely not informative or likely sort of junk. And I'll get more into quality filtering information later, and we can kind of go back and rethink about this 200 genes for each cell, but that's kind of what we're going to work with for now. So we have the current Seurat object function. We're going to tell it where the sparse matrix is that we created above. That's just example.sparse.mat. We're going to name it. We can name it whatever we want, this object. In terms of like in the Seurat object, you can have a project name. I just gave it the cur sample name, and then those 2 filtering steps that I just mentioned. So we can go ahead and run this, and you can see we now have example.seurat, and it's a large Seurat object that's 11 megabytes of memory. Okay, so we can go ahead and kind of look at the Seurat object, this example 1, and kind of get a sense of what it has. I showed that diagram earlier, but let's actually take a look. So we can access the metadata using the at syntax, at metadata, and then I'm just piping that data frame. It's a data frame, I'm piping it to head. And we can just kind of take a look at what is filled out by default. So again, on the rows of this metadata are the cells, and the columns are sort of these just metrics that we've already captured. We can see orig ident is actually the name. It's by default the project name that we defined earlier. That's useful because if we end up combining multiple Seurat objects later, we can still re-identify which sample they came from. N count RNA, it's a little bit cut off there, is the total UMI count. N feature RNA is the total gene count for that cell. The rows are cells. Yep, the rows are cells, and the row names are actually the actual barcode sequence for that cell. Okay, I wanted to mention there's a slot in Seurat called active ident. This can trip you up, I would say. So this is maybe a word to the wise. I've been tripped up by this before. In Seurat, you can have multiple identities for cells. So the current or orig ident is like the sample that it came from currently. But you can imagine that we might have later on, we might have like identified the cell types for every cell. So we might know, these cells are T cells, these cells are, CD4 positive, CD8 positive, et cetera. So you can have a different metadata column define the active ident. And what that does later on is actually Seurat will kind of by default use the active ident as a grouping variable. And if you're not aware of that, you might process the Seurat object in a way that's by group, but it might not be the identity that you thought it was. So it's very nice to know that you can always check this active ident and you can redefine what it is. So right now, if we look at it, it's just for every cell, it's just the sample name that we defined. I mentioned Seurat can have different assays. So an assay, and I guess I probably should have had this as a slide, but that slide I showed with the figure where it was counts, scale counts, or data, scale data, that is actually one assay that can be like replicated as many times as you want for different assays. You can imagine it could either be like a gene expression assay. You might have a TCR assay that's linked to the same cells. In addition, the normalization step we'll use actually creates a new separate assay. And so those assays can be just like idents, they can be defined as like the current active assay. Also useful to know because Seurat by default performs a function on the current active assay. So we can kind of look and, the current active one is RNA. That's actually the only one we have right now. And then within RNA, we can see that we have 14,359 features, 337 cells. There's only a single layer within that assay, counts. That's the raw counts data that we saw earlier. And then you can actually access the raw count data if you go, at assays, pick the RNA assay, and then you can just pick the counts file, and you can see that it's basically it's the sparse matrix that we just created in the first step previously. So a Seurat object is kind of a fancy way of just storing the sparse matrix and a bunch of related metadata. Some other slots that are useful, I think I mentioned these already, so I won't kind of rehash them in depth. There's reductions, graphs, and images. They can store reduced dimensional data, nearest neighbor graphs, and slide images if you want to. Okay, so that brings us to exercise 1. And I recognize that this exercise might be a little bit challenging depending on your level of R. So I will be showing a solution using lapply. And so if you don't get, if you don't get there, don't feel bad. We'll kind of go over the solution and we'll cover it. But yeah, I thought it'd be kind of helpful and nice to have an interactive exercise here. So if you want to just please write a loop to load the raw data for all the samples as Seurat objects into an R list called Seurat.list. And I think you should be able to combine sort of the code from the 2 previous steps that we covered before, and that should get you started. So why don't we pause for just a couple minutes, and I'm happy to answer questions if you guys want to give it a shot and have any questions, or if anybody on Zoom has any questions, please feel free to speak up or put in the chat. I'll just put a timer for like maybe 3 minutes or so, and we can kind of restart back. Okay, so that was 3 minutes. Do you guys need any more time to try to work on it, or you want to look at a solution? Okay, well, let's go ahead and take a look at one way of doing it. So I should mention, in the notebooks directory, there's a subdirectory called keys, and that has a full version of the notebook, which includes the exercises filled out with solutions. And that actually will knit successfully because it has kind of all the code present. So in case you want to kind of take you can find the solutions there, and then you can also knit that whole thing if you're interested in doing that. Okay, so I'm just going to copy this over now. All right, so my solution probably looks large and verbose, partly because it's commented a lot. But essentially what my solution did was combine those two steps inside of a loop. So what I did was loop over the sample names like I had done previously. Again, function can be with a single variable called cur_samp or whatever you want. And the first step could be to create a sparse matrix. And you could kind of point that function to the data directory by giving it file path with this cur_samp variable piped into it or put into it. That would kind of vary the path depending on which sample you're currently looping over. And then that would create a current sparse matrix object within the loop. And then you could actually use that cur_sparse matrix object within the loop in a call to create Seurat object. Just say counts equal to that kind of temporary current sparse matrix. You could define the project name as the current sample. That would store orig ident as the kind of the current sample name that you're looping over. And then you probably or almost certainly want to apply the same filtering cutoff values for every Seurat object. And so that creates a object called cur_Seurat, which is a Seurat object that lives only within the loop. You can return that object from the loop with this return function, and that basically stores a list of Seurat objects. So I'll go ahead and run this. And please, if you didn't sort of come up with the answer, just copy over the key to this code chunk and run it. And that way we can all have sort of the standardized Seurat.list object. And it should take a little while for it to run. Okay. Were you guys able to create the list? Okay, great. Okay, so we can move on. We can do a sanity check just to make sure that our list of Seurat objects is reasonable. In this case, I'm just using lapply again. I'm looping over the Seurat list this time. So that will loop over each of those Seurat objects in the list. And I just create kind of a little test data frame here where the first column is the unique sample. The second column is the number of genes, which we can get using n_row. N_row is kind of just a quick way of getting number of genes, the number of rows in the sparse matrix. Num cells is just the number of columns in that Seurat object. And we end the loop here. And in this case, I'm actually piping the resulting list from the loop using dplyr syntax to this function called doCall. And within doCall, I'm saying bind rows, which is actually another dplyr function that basically just concatenates rows of a data frame. Rows into a single data frame. So that's one way of actually kind of collapsing a list into a single object. You're basically collapsing it using whatever function you define there. So that'll basically collapse the list into a single data frame by binding the rows together. So if we run that really quick, we can see that it gives us a single data frame, 6 rows, and kind of gives us just a nice summary of the total number of genes and total number of cells for each of the samples that we have. Yeah, and at this point, I think it would probably be good, you might want to compare this output to, for example, the cell ranger output, the HTML summary that we looked at, or that you guys looked at with Josh, just as sort of like a QC check to make sure everything loaded correctly. Okay, So moving on, we can cover some of the basic Seurat functionality before we sort of dive in. So the goal here is to illustrate and practice some of the key functions of Seurat. We'll use the example.Seurat object for this, and that corresponds to the first sample in our list. So just kind of keep in mind that this is just the first sample. So the first thing we can do is slice the Seurat object to extract specific cells or genes. So we can slice by cells. First, let's cover that, and we can kind of slice it by expression. So for example, we might want to find a subset of cells based on the expression of specific genes. So this example, I think, is selecting only cells with non-zero counts for the CD4 gene, excuse me. And so what we do is we take our Seurat object, in this case, example.Seurat, we can pipe it to the subset function, which is actually a Seurat version of, it kind of looks like it's the base R subset function, but it's actually going to be applying the Seurat version of it. The way that you can subset is you pick the slot that you want to use. In this case, the only slot we have is counts. I suppose that should be actually layers, according to the new Seurat version. And then the condition that we want to subset on is specified by the subset equals. So this has to be like a logical output with whatever's after the equal sign. And it can be obviously like several conditions that you want to meet. In this case, we're just saying the CD4. So the CD4 gene is just, you can specify by saying CD4 and it's greater than 0. So that'll give cells that have CD4 expressed at all. Yeah. Yeah, Seurat is an interesting syntax sometimes. I don't always agree with how they named things. So this returns, you can see, if you remember, this is actually the same number of genes or features, but I think before we had 377 cells, we now have 142 cells within this assay. Another weird Seurat thing is that when you look at this summary, it says 142 samples. I don't know why they don't just say cells or like, yeah, I don't know. Samples is kind of a weird way to call it, but anyways, that's a nitpicky thing. So now we have, oh, it looks like I changed this. Probably needed to update. 142 cells and all the original 14,000 features. Okay, so we can also select cells by columns in the metadata slot. So for example, total UMI counts. So this is slicing sort of on a different condition, but very similar syntax, right? So we're just calling one of the column names from the metadata. We're saying we want cells with UMI counts, total UMI counts of greater than 4,000. And that gives us 284 cells. Okay, so we can also extract specific genes. So before we were slicing to specific columns or cells, now we're slicing to specific rows of the count matrix. So we do that with fetch data. It's a Seurat object. We can pipe our Seurat, it's a Seurat function. We can pipe our Seurat object to it and say that we want specific variables by this vars element. In this case, I just picked 3 genes that may or may not be interesting. And we want to select it from the counts layer. And I guess I'll give a reminder at this point that since we're not specifying the assay, it's going to be using whatever assay is set as the active assay. So if we had like RNA plus scRNA-Seq, whatever the active assay is at this point, it's going to assume that's what we want. So always be careful of that. And then looks like I'm piping this to slice head, and then so that'll give us the first 10 cells worth of counts for those 3 genes. And then don't worry about what this is doing. This is kind of just making a nice table. Honestly, just for a solution for this RStudio visual mode not having great ability to show tables. So that ends up giving us this table where the rows are now cells, and the columns are those genes that we chose, and the values are the expression of those genes. So we can do both. We can slice genes and cells. So we can imagine we might want counts for the CD4 gene among cells that express it. For example, we could kind of concatenate those two functions that we just saw. So first we could subset cells to where only cells that have CD4 expressed. And then next we could fetch just the CD4 gene, counts for that CD4 gene, and then kind of pipe that to slice head and take a look at the first 10 rows of it. And this should basically be the first 10 cells that express CD4 at all, and then the counts for the CD4 gene among them.
So those counts are like in that cell, how many, like, what are those counts actually?
Yeah, so those counts are the UMI counts for the CD4 gene.
So how many transcripts of that gene are there?
Yes, how many transcripts? Yep, pretty much. Yep. Okay, any questions about subsetting? Any questions on Zoom? Okay. Okay, so we can move on to plotting metadata. So I think there's a couple of two main plots that Seurat kind of commonly shows, although you could do other things. There's this violin plot, which you can do by this function VLN plot. It's pretty straightforward. You just give it a Seurat object and you specify the features that you want to show. Typically, these are metadata columns from the metadata slot. In this case, I'm using the total counts, total UMI counts, and the number of features or genes. And it's going to basically show us, well, I'll go ahead and run it. These are, if you've never seen a violin plot before, it's kind of like a mirrored density plot that's rotated 90 degrees. So you can imagine, like, you know, a density plot is kind of like a histogram. It's like a smooth histogram. Take that density plot, rotate it 90 to be vertical, and then mirror it kind of about its own x-axis, I guess y-axis. And so what you see is basically the distribution of cells. Every dot on this figure is a cell, and you can kind of visualize the distribution of each of the variables that you picked for this current sample. And I think by default, it colors by sample if you had multiple of them, but in this case, we just have a single one. So you can see we have sort of a cluster of cells for a number of total UMI counts. We have a cluster of cells, you know, down here around 5,000. We have some kind of outliers on the lower part of the distribution that have very few UMIs counts. And then we have some outliers way up here that have, you know, 5 times kind of this 5 times the number of counts as this bulk distribution down here. A similar story with the total number of genes. We kind of see this big cloud down here at the bottom with some tails at either end. So we'll use these to basically look at QC metrics when we're doing some filtering, and yeah, that's kind of the main purpose of them. Another kind of QC metric is, or visualization tool, is the scatter plot function. So you can basically make a scatter plot on 2 features that you choose from the metadata. In this case, we're using the same 2 features as before. It's going to basically draw a scatter plot where every dot is a cell. And you can, you know, very clearly see there's a very large relationship between, or a very strong relationship between the number of total UMIs and the number of genes or features that you pick up in that cell. And it gives you, I think this is probably a Pearson correlation up here.
Then that's the case
I think for 10x data, people often find that strong relationship. Yeah. I mean, it kind of makes sense, right? Because like the more total UMIs you see is kind of indicative of how much sequencing you've done in that cell. And the more sequencing you do in that cell, the more genes are likely to have at least one count from it. If you have a small amount of sequencing, you might miss genes. So I think that's kind of, you know, we bring up is like a bit illustrative of that dropout phenomenon I was mentioning earlier, where you just don't see genes sometimes that are expressed at all in cells, where you would expect them to be expressed. And as a side note, I think Josh and I were looking at the Cell Ranger HTML file, and I won't pull it up here, but I think they do have on there a graph of sequencing. What do they call it? I think it's like sequencing effort or saturation, I think, is what they use. So it's sort of like a curve that tries to indicate, whether you've sequenced deeply enough to sort of have a good idea of, the number of features that might be present. Okay. So these Seurat functions are nice for visualization, but they are kind of a little bit limited, to be honest, in what you can do if you have sort of some customization you need. They are based on ggplot functions, but it's kind of hard sometimes to modify them. So I did want to point out, and actually this is going to be a brief exercise, that you can also use ggplot to plot things. So why don't we break for a little bit, and the exercise is to please write code that generates a density plot of the total UMI counts for that example Seurat object using ggplot instead of using Seurat. And I guess I'll give a hint that, the total UMI counts are found in that metadata slot of the Seurat object. So that should maybe help out. Okay, so I'll give you guys just a few minutes here. We can, yeah, I guess we can pick back up here. Were you guys able to plot that custom ggplot function? Okay, great. So yeah, we can take a look at, I think my solution is probably as good as any. Pretty straightforward here. I think what I did was just take the metadata, slot using the syntax at metadata, pipe it to ggplot, set the total number of UMI counts using this aesthetic function as the x-axis, and then you can use geom density to create a density plot, and I just filled it with blue. That, of course, is going to look like this, which is pretty much half of the violin plot, on a 90-degree angle. Okay, great. So this is obviously like a fairly easy exercise, but you could imagine that if you had something more complex that you wanted to do, if there was another ggplot function that was, useful for you, I can imagine, or I think I've used, where like you might want to do a Venn diagram, or something like that, where GG Venn diagram is a separate function or separate package. You wouldn't be able to do that with built-in Seurat things. So this is just an example of how you can do those kinds of things. Okay, so you can also modify metadata. That's a pretty useful and common thing to do, and it's pretty easy because the metadata object within Seurat is a data frame like any other data frame. So here's some just sort of two basic examples of creating a new metadata column in the data frame, in the metadata slot. So this first example is just me creating a dummy column where it's just going to fill every row with test.metadata, this character string. A more useful kind of thing to do might be to create an indicator for cells with high UMI counts. So in this case, I'm just using if else based on the total UMI count column being greater than 10,000 and returning true if it is greater than 10,000 and false if not. So yeah, pretty easy to create new metadata. You can see what that looks like here. And so this brings us to something that we need to do for our data set. Because we have, if you recall, we have 3 samples that have Alzheimer's diagnoses and 3 that don't. We really actually need to kind of have that metadata in our Seurat object in order to do some downstream things. So I wanted to have that be an exercise of we'll add that metadata to every Seurat object in our Seurat list. Before we get to that exercise, I just wanted to share that the metadata is, I pulled metadata from that sequence repository, and I put it into a CSV file previously. And so the path is defined here under this list files. It's kind of boring what this does. It basically just finds any file that meets this pattern with metadata in the file name and gives a full path to it. So that should be a path to the metadata. It's a metadata file. It's a CSV file. And then here I'm just doing well, first I'm reading that CSV file using the path that we just defined. There's a couple of just data cleanup things I'm doing here. Selecting, there's a lot of metadata columns that we don't need, so I'm just selecting away some of them. So if we run that and take a look at it, you can see it's a data frame, 6 rows, 4 columns. Importantly, it has the sample names, which are the sample names, defined in our Seurat objects within the list. It has the diagnosis information, has age, has sex. As a side note, you can see I picked only female patients. That's mostly just to limit the variation of gender that might include. Okay, so exercise 3, why don't you guys go ahead and try to write code that adds the metadata from this CSV, SRA.metadata, to the corresponding Seurat object in Seurat.list. And I will give you a hint that inner join is probably very useful in this case, dplyr function. Okay, so I'll give you maybe 4 minutes. Okay, so what I did to do this was, again, looping over the Seurat objects in the list. As before, cur_samp, although now with a period apparently, is the current Seurat object. And what you can do is actually just use inner join with the Seurat object metadata and the SRA metadata data frame, which is in the global environment. And you can just join using these columns that are named differently but identical between the two. So essentially, this is like a one-to-many join where SRA metadata, if you recall, has just 6 rows, and cur_samp metadata, I won't do it here, but it could have the same number of rows as the number of cells there are in that Seurat object, and that orig ident is the same sample name basically as here. So when you do inner join, it's just a one-to-many join, and it just adds the metadata basically to the metadata in SRA.metadata to the Seurat object. So if you just want to copy over that code from the key and run it, then we can all have our metadata present. And then, as I usually like to do, and I would recommend doing when you're doing workflows like this, is including checks to make sure everything is going how you think it's going. So I have just a check here of the metadata. Essentially what this does, it's wrapped in try catch. And so if it fails, it'll give you a message. What it's basically trying to do is it's trying to do a function that will only succeed if all of those metadata columns from the SRA metadata file are included in the Seurat object, and it's doing that for just this first object. And if they're not there, it'll fail and it'll give you this message. So success, we have all of our metadata into the Seurat object, at least for the first one. Again, I just as a piece of advice, someone who's been programming for a while, these kind of checks are really great because if your data changes or something changes upstream and you didn't realize that you're not loading the right metadata or whatever, this check will hopefully catch it. Something that you want to do. Okay. So that takes care of code section 1. Were there any questions or lingering thoughts before we move on? Okay.
I have a question.
Yes.
Is the try-catch function, is it only in this package, or are there similar functions in other packages, just so that if we're working in others, we can kind of use similar methods for catching problems?
Yeah, absolutely. Very good question. Try-catch is a base R function, so it can be used for pretty much anything. And I am not an expert in try-catch. I don't often use it for necessarily for catching errors in this way, although I did use it here. It essentially, it allows you to run any R function that might fail and give you an error essentially what it does is keeps going with that function and just reports the errors to you. So in this case, either way, it was going to report a message, whether it was going to be success, you did it, or failure, here's a message for the failure. But yes, you could use this for checking, a lot of things. And I think there are, probably other standard ways of testing. I often will check, it depends on what I'm doing. I'll check to make sure something's present, a column is present, or number of cells look like they should. But yeah, it's a great habit to get into, I would say, in general, not necessarily requiring try-catch, but try-catch is a good tool for sure. Okay. Okay, so why don't we just take a quick 5-minute break here, and we'll reconvene in just a little while. Okay, all right, so I think we're all powered up and ready to go here. So we can resume. So the next topic I wanted to talk about is removing multiplets. You'll hear me kind of say doublets and multiplets interchangeably. I think mostly we're referring to doublets when we talk about this. Okay, so doublets and multiplets. We've seen this before, this figure. This is when we're creating the emulsion droplets. We know that sometimes we get empty droplets by design, but what we really want to avoid is actually multiple cells being placed into the same droplet. And that's sort of illustrated here by this gel bead E droplet. Of course, there are 2 cells within that. And that's problematic because we can't distinguish between the molecules that we extract from those 2 cells. They'll have basically the same cell barcode, and we'll kind of have this confounding effect. I will note that there are both heterotypic and homotypic doublets that people talk about. So homotypic doublets are where 2 cells of the same type are placed into a single droplet. And the effect of that is, according to the literature that I've read, relatively innocuous to the downstream analyses. If you have 2 of the same cell type, obviously there's cell-to-cell heterogeneity in gene expression, but by and large, you'll have similar gene expression profiles. And so when you add those together, you don't have sort of a huge error, the same way that you might have heterotypic doublets, which would be where 2 cells of different cell types are placed into the same gem well. That is a much worse situation where you have this confounding effect of cell type by expression. So we try our best to basically remove those doublets or multiplets from occurring. And we're going to be focusing on the heterotypic kind. I did also want to mention that there are these things called barcode multiplets, and that's actually illustrated here with the purple gel bead B. Sometimes, and I think this is obviously tried, they try to minimize this, but sometimes 2 beads will actually have the same cell barcode. And so if each of those beads, each of those droplets gets a cell encapsulated into it, it will appear downstream like there was a doublet, because if those 2 are different cells, we can't distinguish those 2 droplets from each other. That doesn't occur super often, but it is kind of another kind of mechanism for how doublets can happen. Okay, so along with many other steps, there are many tools to remove doublets. I'm focusing here on this one called SC Double Finder. That's what I'm going to call it. And it is a recent tool from Germain et al. 2022. Basically what they did was combine steps or methods from several previously published methods into like a kind of super method, so to speak. Like it does the method, it does what 3 methods have done previously. So I'll kind of walk through what it's doing. It's pretty complicated. I won't go into all the details. In fact, I'm going to gloss over a lot of it. It's also a little bit confusing because in the process of doing this, we're actually doing a lot of the downstream steps that we will cover like even next week. So for example, like dimension reduction and clustering, it's actually doing that kind of right now in the course of identifying the doublet. So it's a little bit kind of confusing in that way, but we can walk through it. Okay, so the first step, what it does is reduces dimension of the gene matrix, and it clusters the original cells. So the dimension reduction that it uses is singular value decomposition. I won't go into what that does, but suffice it to say that you can kind of compress the count matrix into fewer dimensions and maintain a lot of the same information. It then creates this k nearest neighbor network, which then is input into the Louvain algorithm. I'll actually cover more detail what those do, and actually I'll cover Leiden algorithm, but they're kind of doing a similar thing as Louvain. It's basically just a way of seeing in this reduced dimension space how closely related cells are to each other with respect to gene expression in creating this network. And then the Louvain algorithm actually tries to cluster by minimizing this thing called modularity index. So suffice it to say that Louvain is clustering cells by gene expression. And so as output, you kind of get this figure on the right where individual dots are cells and they are clustered in this reduced dimension gene expression space. And the colors represent what the Louvain algorithm considers to be kind of distinct clusters. So then what the algorithm does is actually generate artificial doublets. And so what it does is it randomly selects pairs of droplets from two separate clusters. So that's creating these heterotypic doublets that I mentioned previously. Those are the ones that are of interest mainly in removing because they are most problematic. So for 75% of these pairs, it creates a doublet by summing their counts. So it literally just takes their counts across, for all the genes, and it just sums them together. So that kind of looks like a doublet, right? Sort of an artificial way of creating what would look like a doublet. For the remaining 25%, they sample from a Poisson distribution using the sum. So just kind of a different way of generating these doublets in somewhat more of a stochastic way. And then they will again do dimension reduction and clustering, but this time they actually use a mixture of the original cells and the artificial doublets using the same kind of methods I described before. And you can see in this figure, they have the original cells in red and the doublets in blue, kind of illustrating that the doublets do sort of sometimes cluster apart, sometimes don't cluster apart. And from that, then they construct this k nearest neighbor network using this mixture of doublets and original cells. And again, I guess it should be obvious, but in this process, like we know which cells are doublets, the artificial doublets, so we can recover that information later. Okay, so then they essentially using this k nearest neighbor network and a couple of other data points or variables, I guess, from the overall data set, they derive these cell level predictors. So from the nearest neighbor network, they find the proportion of nearest neighbors that are artificial doublets for every cell, every original cell. And they do that for different values of k. So k is like how many nearest neighbors you're looking for in order to define the neighborhood. And so for multiple values of k, they find this proportion. They also do a PCA dimension reduction principal component. I am going to gloss over that for now. I'll talk about it a little bit more next week. But they do use a couple of the PCA components from that dimension reduction as predictors. They use total counts, total number of features, and then a co-expression score, which is basically one of the other previous methods that were published. So this is their way of incorporating that method. Essentially, what that method did was try to identify doublets based on the premise that if a cell co-expresses multiple genes that are usually mutually exclusive, then it's likely a heterotypic doublet. So that tool would actually look at these this list of genes that are supposedly exclusive. You wouldn't see them expressed in the same cell, and it would kind of decide whether that was a doublet or not. So that co-expression score gets used as one of many predictors for the downstream step. So then using those predictors, they train a boosted tree-based classifier. I'm going to gloss over how that works. It's basically a classification method that is going to try to estimate a probability for whether every original cell is a doublet or not. And one of the outputs is a score, and that score can be interpreted according to them as a probability. And so their final step is to, from that raw score, which is a probability, decide a threshold score for classifying a cell as a doublet or not. And they have kind of an algorithm. They came up with this kind of a distinct cost function that they try to minimize using this algorithm. That cost function includes the misclassification rate in terms of false positive and false negative. And it also includes this deviance from the expected doublet rate. So this cost function is sort of like using several different components. They minimize it, and this figure on the right actually shows the cost function in black, the false negative rate in red, and the false positive rate in blue. And then that deviance from the expected doublet rate is in gray. So the cost function sort of comprises all three of those and in a specific way. And then they try to minimize that. And in this case, it looks like they decided a threshold of 0.475 was optimal for this example. So then, yeah, once the threshold is determined, then any cell that meets that threshold or exceeds it will then be called a doublet in the algorithm. They claim, and this was verified in addition to the authors claiming it was verified by a separate publication, which I put on the bottom, that SC double finder is fast and accurate. These are each of these panels is showing sort of the results for two separate artificially created data sets that have doublets included in them. You can see SC double finders in the yellow, so it's on the left, it performs better in terms of true positive rate and false positive rate. And on the right, it performs kind of on par with the other methods. So it seems like it's pretty good. It is more recent than the other one, so I think that's nice, and it's pretty easy to implement. Okay, so are there any questions on that before we move on to the code? So we can move on to code section 2, removing multiplets. Okay, so this is kind of typical of experiments or workflows that I've done in the past. This particular package and tool is actually not available for Seurat, or at least it doesn't work with a Seurat object. It's actually part of the bioconductor kind of ecosystem of packages in R. So in order to use it, we actually have to convert our Seurat object to the data object of bioconductor, which is single cell experiment. So that'll be the first step that we do. Again, I've had to do this before for other tools. I think it's something that you probably want to consider fairly normal. Okay, so the first step is we can actually just convert the Seurat object to single cell experiment using the Seurat function as a single cell experiment. We just give it our example.seurat, which again, that's just the first sample. It's not the full list. And we indicate which assay we want to port over to the single cell experiment. In this case, it's the RNA. It's the only one we have. Below is basically doing that same thing, except for all of the Seurat objects. So this will just make a new list of single cell experiment objects. It does give us this warning here: the data layer is empty and scale data is empty. We knew that. We haven't done any transformations or any scaling on our raw counts, so that's fine. It's just empty, and that's what we expect. Okay, so this is an example of the checking that I would normally do in such a case as this, right? So like we've converted from one object to another, and everything seemingly has gone fine. Maybe this is me being paranoid, but I just want to make sure that everything has actually gone fine in most cases, or usually in all cases where I'm doing something like this. So what I have here is code that actually checks a couple of different things between the two object types. And it does it for all of the objects in the list. And basically it's going to return, if it checks them, if they're all true, then it's good. If any of them is false, then it actually throws an error using the stop function. So I'll just kind of walk through this quickly. The first one is we're actually checking that the count matrices match exactly. There's this all.equal function in R that will match, basically check if two objects are nearly identical up to like machine precision, which can't be exactly identical. Then we check that the values of the total UMI counts match, check that the values of the total number of genes match, and then check that all cell identities match. And that's all wrapped in this all function, and all what it does is takes any number of logical elements, like a vector of Boolean values, and if they're all true, then it returns true itself. So if all is true, then we get this message, the metadata and data match. If it's not all true, then we get this error. So we can run this and see that for all 6 samples, it looks like everything matches. So should be all good. I'm sure the authors of as.Seurat have checked, or sorry, as.single cell experiment have checked this extensively, but I always feel better sort of doing these checks throughout. Okay, so now we're ready to apply SC double finder to identify the doublets. The function actually returns a new single cell experiment object with additional data, actually additional metadata, so we can overwrite the existing object, very similar to how Seurat functions work. And I believe, yeah, okay. So if you open the console, you can actually see the output as it's going on. But basically, it did the steps that I mentioned earlier. It created 1,500 artificial doublets, did dimension reduction, k-nearest neighbor network. It trained this boosting tree model that is a classifier model to identify doublets versus not doublets. And it does this iterative training, so that actually has to do with the threshold value that it's finding. It reports that threshold value that it found as 0.475, and then ultimately it found 7 doublets. And I think this sample had 377 cells. So yeah, it found 2% were doublets. So the doublet identities are stored as a new factor variable in the metadata called scdoublefinder.class. We can find that. So I know single cell experiment is a different object type. I'm not going to go over where everything is, but the metadata is actually in this @call data slot. You have to coerce it to a data frame, and then you can kind of do some standard dplyr to it. This is kind of just what that first 6 rows look like. These are individual cells on the rows, and we can see that SC double finder found that all 6 were singlets. Yeah, so then we can go ahead and apply SC double finder to all of the single cell experiment objects in our list, and it will take a little while to run all of them, so we can just kind of wait for it. And are there any questions while we're waiting? I guess one comment I've noticed running this repeatedly is that it doesn't always find the same number of doublets each time. I don't know if it's a seed thing or what, but yeah, maybe something to keep in mind. I think you can, and I think it also mentions that it sets a seed of something by default, so I don't know why it would come up with different numbers, but I didn't look into like maybe somebody's brought this up on their GitHub, repo as an issue. I'm not sure. looks like it's finished here. And you can see that it has called a different number of doublets and even a different percentage of doublets for each of the samples. And I think I have some code here to loop over and report how many doublets it's found by sample. So yeah, differing number and differing percentages. So then the next step is, now that we've identified the doublets, we can filter them out of the single cell experiment, and then we can convert that back to the Seurat object for downstream purposes. So the way that we subset a single cell experiment object is actually a similar way that we can subset a Seurat object, although I haven't shown it yet, which is, if you're familiar with base R, not sure how many people are familiar with base R subsetting anymore, but you can subset things using these square brackets. And the syntax works like, you put the object first that you want to subset, you put a square bracket, and you can subset rows by putting conditions before a comma, and you can subset columns by putting conditions after a comma. If you put nothing, then it assumes that you want all of one of those elements. So in this case, we're saying we want all the rows from the object, which would correspond to all of the genes. And then we have this condition for the columns that we want the sc double finder.class to be singlet. So we're going to remove anything that wasn't called as a singlet. That will actually give us a updated single cell experiment object with fewer cells, only the ones that were singlets. And then we can actually pipe that to the Seurat function as .Seurat and basically convert it back to a Seurat object. And in that function, we tell it that the data layer should be empty since we haven't done any scaling. And we want to say that the count layer in the Seurat object should be filled with the corresponding counts layer in the single cell experiment object. All right, so that created a new Seurat object from our single cell experiment. And I think what I'm doing here is just comparing the original Seurat object with the filtered Seurat object. And we can see the original had 337, the filtered had 330, so we filtered out 7. And we can check that against our output for that example, which we can see did actually indeed filter out 7 doublets. Okay, so that brings us to exercise 4, which is if you could please write some code that filters out the doublets from each of the samples and returns a new list of filtered Seurat objects called seurat.list.filt. And I think this should be fairly straightforward to do with a loop, but I'll just give you guys a couple minutes to work on it and reconvene. Were you guys able to kind of filter them into a new list of Seurat objects? Okay, great. Were there any questions, or I guess we can just go over what I had here. Okay, so what I did was take our SCE list of single cell experiment objects, loop over them, and basically do that same subsetting task, except this time, using the looped looped object, which would then filter and return a single cell object, and then basically just pipe that to Seurat, and that will return a list of Seurat objects that are filtered. So let me just go ahead and run that. Is that kind of similar to what you guys had done? Yeah. All right, great. Okay, I have a check here. Basically just loops over the Seurat, actually looping over just a vector of 1 to the length of Seurat list, which would basically just be like 1 to 6. The reason I did that was because I want to actually compare the 2 Seurat objects. So you can see that kind of pasting this message down here for every individual project or Seurat object. But I'm comparing like the ith Seurat, original Seurat list to the ith filtered Seurat list. That's under the assumption that they were, processed in the same order, which they were, but that basically just will provide a message for like the difference in the number of cells, which is basically the number of cells that were filtered out. So it looks like it was, 6, 90, 65, etc. So those should hopefully match up to the number of doublets that were found, which it does.
So you have to use as-applied here because you're comparing two list objects, right? So you need like a third object that is a vector to compare them within, right? Or you could do it with lapply if you then created a list that encompassed both.
You actually can do it with lapply either way. I think, I could be wrong, but like if I just change this to lapply, I'm pretty sure it'll run just the same and it'll just return a list of those, right? lapply, like they're both looping. It's kind of just like what it returns.
So in this case, you get a list, and in as-applied, you get that.
As-apply, like as far as I understand, it's like really just a simple output version of lapply, where like it'll try to coerce the list automatically to a vector for you. It'll actually coerce a data frame for you as well if you try to sapply and return a data frame instead of like this is just a character string that it's returning. Although I found that it doesn't always get the transposition right. Like sometimes it'll return the data frame transposed to what I would expect or want. But yeah, here I like sapply when I'm just returning a vector of things. Yeah. Yep. Good point though. Yeah, good question. Okay, great. That covers that. So let's see if we can cover section 3, filtering cells and genes. And we may or may not have time to finish that. So just maybe a fair warning that we may have to kind of pick up there next week. Okay. So our next step on the workflow with samples still separated is to filter cells and genes. So really the goal here is to remove low quality or otherwise aberrant cells. The people who created the SC double finder have found, according to them, that there's actually low overlap between doublets and cells that are filtered out by QC metrics. So I think previously I've seen a lot of workflows where the doublets are attempted to be filtered just using the QC metrics, and you sort of do both in the same step. It seems like they've found through their, they've done some pretty extensive testing in their papers, and you probably do want to do both. So I would throw that out there. The strategy basically is to remove outlying cells according to some key quality control metrics. I wouldn't say that there are universally defined or agreed upon metrics, but there are kind of some recommendations that seem to come across from different papers I've read. One of them is that the metrics often differ by samples, and so you want to be defining the outliers sample-wise. I think previously I've seen where you might filter on a global metric. You might just say like, the percentage of X thing can only be 10%, and I only, anything above that, I'm going to throw it away. I think it's much better to actually define the outliers by sample. Another good strategy that I've seen is to define outliers dynamically, or in other words, define the threshold that excludes the outliers dynamically using like the empirical distribution of the metrics. That's actually what we're going to do here. We're going to look at the distribution of the quality control metrics, and we're going to filter based on, some measurements of the spread of that. Again, that's like sort of in contrast to, you can imagine just saying like a hard cutoff, like no matter what, I'm just going to use this hard cutoff value. And then the other thing is that the metrics are pretty well understood to differ by subpopulation of cells. So, right, like T cells might have different metrics from B cells, or et cetera. So the recommendation there is actually to filter jointly. What that does is kind of prevents you from excluding truly good cells that meet maybe just one or two of these quality metrics. Like, let's say you have some subgroup that is a larger number. Sorry, the cells are actually larger. They might contain literally more, more RNA within them. So your total counts would be higher for those larger cells. So those might meet the threshold for that like total UMI count, but they might not meet the threshold for other metrics. So if you just use individual metrics, you might throw away good cells in that case. So I think people have found, and Lucan and Feast talk about this in 2019, filtering jointly by the metrics. And then, yeah, there are no a priori thresholds. I think people generally start with permissive values, and they generally examine the downstream clustering. So like getting down to the UMAP, and they'll adjust as needed, whether they find, clusters of cells that seem like they're weird or they can't characterize that cluster. Those might be some extraneous cells that they should filter out. So they might go back and, modify their filtering criteria and then kind of readjust. Germain et al. Talk about this like Goldilocks filtering approach where, you don't want too strict of values because you might throw away good cells. You also, you want to do some quality filtering because they're in most cases are kind of bad quality cells that you want to remove. All right, so here are the metrics we're going to use for today. Again, I want to stress that this is not like a universal truth. Like these are not the only metrics you could filter on. These are not the only values you could use for them, but these were adapted from Germain et al. 2021. And I guess I should mention I have these references at the end of the slides in case you want to look at them. So what their kind of method is going to do is we're going to look at these 4 metrics, which are log total counts or UMIs, log 10 total features, percentage of counts that are in the top 20 highly expressed features, and the percentage of mitochondrial counts. Or percentage, like more precisely, percentages of UMIs that map to mitochondrial genes. The rationale for some of these is like basically empty droplets or multiplets, so empty droplets in the sense of like if you have a low total counts, those might be empty. If you have a very high total counts, those might be multiplets. And then for mitochondrial counts, usually that's indicative of like a broken or lysed cell that was like, dying or going through apoptosis during the library creation step. People look at percent mitochondrial because if the cell breaks and the cytoplasm is removed, the actual mitochondrial regions will be like overexpressed because the messenger RNA has leaked out of the cytoplasm. Okay, and then the way we're going to filter on these metrics is basically using median absolute deviation. So this is sort of like an alternative to using standard deviations and means. We're going to use medians and median absolute deviations. It's sort of like I guess, less prone to outliers. Looking at these median absolute deviations. So I think that's the recommendation. And then we'll use the cutoffs that Germain et al. Use in their paper. So just to take the first example here, we're only going to keep cells that have a lower limit of total counts that's greater than five median absolute deviations below the median. So like you can imagine like the median, 5 median absolute deviations below that. If there's any cells below that threshold, we're gonna exclude them. And then similarly for the upper limit, we're gonna find the median of the log 10 total counts. We're gonna find 2.5 times the median absolute deviation from that median. And then anything above that cutoff, we're gonna exclude. Okay. So we can also filter genes by type. We're moving from filtering cells to filtering genes. So in addition to filtering cells, we can also filter genes if we want. This is one of those things where it's sort of up to you and up to your research question. And also I think it's somewhat subjective. I think there are reasons for filtering and for not filtering. I'll go through the rationale. So for ribosomal protein messenger RNA, right? Like the ribosomes are they include proteins and actual ribosomal RNA. There are, genes that encode those ribosomal proteins. So our messenger RNA can map to those ribosomal protein genes. Those are among some of the most highly expressed genes among many organisms. So I think the concern or the rationale for excluding them is that they actually can hide other more interesting biological signals. Freitag et al. 2018 kind of gave that as their rationale. I think I would say, do you need to exclude them? Maybe. I think if some cells that you're interested in characterizing are found to be, characterized to some extent by the ribosomal proteins, then they might be useful to have. People have in the past filtered out UMIs that map to mitochondrial messenger RNA. The rationale is basically that they were associated with cell degradation. And I would say probably not needed to exclude those. They're not expressed a ton, and Germain et al. 2021 said they didn't find a whole lot of difference filtering those out versus not filtering them out in terms of downstream processes. Similar story with non-protein coding messenger RNA. So the rationale for excluding those is that they're not well characterized in like gene databases or transcript databases. I actually kind of thought this was interesting. I didn't know this until I looked this up, but of the like 100,000 cDNAs that were cataloged in this Phantom3 database, I think it's a mouse database, like a third of them were actually non-protein coding messenger RNAs. And so, yeah, there's a pretty high proportion actually of messenger RNA that are captured by the 10x assays because they're polyadenylated, so they'll be captured by the assay, but they don't actually code any proteins. I wouldn't say you need to exclude these. Germain et al. Found that they don't really, excluding them doesn't have a huge effect on the downstream analyses, unless you're maybe annoyed by them, but you can always filter them out later. Okay. So we can also filter genes by abundance. We sort of actually already did this. If you recall, when we created the Seurat object, we said we only want genes that are expressed in at least 200 cells. I think this sort of reiterates what I said then, is that these genes are expressed in a small number of cells, so they probably aren't useful necessarily for broadly characterizing the cells, although you might be interested in them. And so you might not want to filter them out at this step. And keep in mind that I guess like filtering out genes that are expressed in fewer than M cells would not let you really characterize any cell populations that are less than M. Okay. Any questions on that? All right, so let's pop over to the code here, marked on file. And I do have a couple of sort of intro things here, pretty much what I just said. Okay, so I did want to mention grep searching. I don't know if that was covered in previous workshops. I'll just kind of give a really brief intro to it, but it is relevant for how we identify percentages of genes. So basically what we're going to do is actually ultimately use this percentage feature set function from Seurat that operates on an underlying grep string search for among the gene names. So you kind of have to know what a grep string search is in order to use it intelligibly. So grep is basically like, it's almost like its own language, regular expressions. It's been around for a long time. It's not unique to R. You can do grep searching like in Linux tools. Maybe you've come across it before, but anyways, it's very powerful for finding pattern match. It's basically pattern matching. So here's an example of basically where we can search for the gene names for any gene that starts with CD4. So you can use this grep function, which is the base R function. You put in a string pattern that you're looking for, and this is where these sort of like, there are these special grep characters that come into play here. This example has this caret sign. Caret's a special character in grep that means start of line. So we're only going to look for this pattern CD4 at the start of a line. This would not return any genes that end, for example, in CD4 or contain CD4 in the middle. And so then we pass it the strings that we're searching in as the second thing. And then this is an optional thing, optional flag for value equals true. If you don't put this, it'll actually give you like an integer, which is the index location where that pattern was found. I think it's much easier to actually return value equals true. So if we run this, we can see that of the full list of genes in this example Seurat object, it looks like we have 7 here that start with CD4. So it came up with CD48, CD46, CD4 itself, CD40, LG. So yeah, so you can see it's like pretty useful for finding sort of a you can use it for like non-exact matching for strings. So another kind of more complicated example here, we can use these square brackets to find basically any of those characters. So in this example, we're looking for, among the gene names, we're looking for it to start with CD. And then any character you put in the bracket, it will search. It's kind of like an OR operator. So you can have CD4 or CD5. And of course, if you have CD45, it'll also find that. You can have CD44, it'll find that. So let's just see what this gives us. So yeah, you can see it found CD52, CD48, CD55, etc. Okay, so those are two like kind of basic ways of doing some grep searching. It's relevant because we can find mitochondrial genes using the fact that the gene names for those mitochondrial genes always start with MT dash. And so it's just the way that the gene names were configured. They're always going to start with MT dash. And so we can kind of look for that exact string. And obviously we want it, the gene name has to start with that for it to be valid. So we're going to basically, in this code, we loop over the filtered Seurat objects. We're defining a new metadata column here called %mt. We're using that Seurat function I referenced earlier called percentage feature set. And we're passing it the Seurat object, and we're telling it a string, a grep pattern to find for those mitochondrial genes. What percentage feature set does is actually finds the genes that match this pattern. It sums the counts for all of those genes that match the pattern, and then it finds the percentage of the total counts of that sum. So we can go ahead and look at what it gave, and yeah, it looks like 3.5 to 5%, depending on the cell you're looking at. Any questions on that? Just a grep searching or what percentage feature set does? Okay. I wanted to throw in looking at ribosomal genes, very similar code, I would say, similar method. Some researchers have filtered out cells based on counts mapping to ribosomal genes. We're not going to do that here, but it's certainly something that people have done. You can basically analogously find %ribo. Ribosomal coding genes always start with RP, and then they have either S or L. So that's kind of where this square bracket comes into play. They'll either start with RPS or RPL. I believe S is for small subunit, L is for large subunit. So this pattern will find any and all ribosomal protein coding genes and give us a percentage. Go ahead and look at it. It's pretty high. So yeah, it does comprise a large number of the total counts. Okay. So before we examine some of these QC metrics, we need to derive them similar to what we've just been doing. So we'll derive log 10 counts, just using the log function base 10, log 10 features, again, log function base 10 of the n feature column in the metadata. And then this percentage of counts that are in the top 20 highly expressed features, much more challenging to accomplish that. So I'll kind of walk through what my code does to do that. I'm not aware of a Seurat object that does this sort of for you. So I had to do it myself, and I'll show you what I did, and hopefully it's maybe useful for you at some point. Okay, so let's work outside in. So we are going to use this function apply. Apply is in the same family of functions we've been using, except apply works on a matrix or a data frame. And the way it works is it loops over either all of the rows or all of the columns of this matrix or data frame that you give it. You decide whether it's rows or columns using this margin parameter. Margin equals 2, loops over the columns. Okay, so that's the first thing. So we're using this apply to loop. And recall that the columns are cells in the Seurat object. So we're basically iterating over the cells in the object. And we are first extracting the counts from the RNA assay, from the count slot for the current Seurat object. And we're looping over that matrix, right? That's a sparse matrix that we're looping over. So then we're defining a function sort of on the fly here for apply. I'm just calling the variable x. Again, x is going to be a column of a matrix, which makes it a vector. So we have a vector called x, and kind of working inside out here, we have a sort function. So we're sorting that vector x in decreasing order. So that's going to give us the all of the feature counts for all the genes for that cell in order. We're subsetting that vector by saying we only want the top 20. So that's basically the top 20 counts for the most highly expressed genes in that cell. We're summing that vector of top 20 to get the total UMI counts that are in the top 20 features. We're then dividing that vector by the sum of x. The sum is the total UMIs for that cell. And then we're multiplying it by 100 to get a percentage. So we do that for every cell that we have in our Seurat object, and we're storing it into the metadata data frame. And of course, the rows in this metadata frame correspond to cells. So the output of our apply function will just be on the cell level, and that should work fine. Okay, so that's a lot in one function, but it's kind of nice when you're looping over things to just take care of everything in one go. Yeah, and as a side note, it's probably possibly a more efficient way of doing this. You could actually parallelize the lapply in a different way, but anyways. Okay, so for a single sample, we can see that if we look at just these, these new quality metrics that we've derived here, we can see we have percent mitochondrial, percent ribosomal, log 10 counts, log 10 features, and our percent top 20 features. So we are ready to visualize, and I see that we have 3 minutes left, so I'll just cover this visualization really quick, and then we can adjourn for today, and we'll pick up at cell filtering. So just really quick, we can loop over our Seurat object that we've created this new metadata for. We'll create a violin plot, which was that same one we saw earlier. The features are just the 4 that we're interested in that I outlined earlier. And n col equals 4 is just going to put them all side by side for us, so it'll be easy to see. So we can go ahead and kind of look at the distribution of the cells according to these 4 metrics across the samples. Okay, so here's sample 1. We can see that for percent mitochondrial, there's these very extreme outliers up at the top here. Obviously, the bulk of the cells are much lower in percentage. Similar for percent top 20 features, there's these really high outliers that have a lot of reads in the top 20 features. And then a kind of an inverse story, I guess, for log 10 features and log 10 counts. You have these outliers at the low end. And yeah, so that kind of summarizes, you can see across all the cells for each of these samples. So basically we're looking to remove these outliers that we can kind of visualize here in these QC metrics. So our kind of approach is going to be removing those outliers using that median absolute deviation method I mentioned, and then we'll kind of go ahead and replot these features, and we'll see how they've changed after filtering. And we hope to see sort of fewer outliers and a more even distribution. Okay, so yeah, I think that pretty much covers it for today. We're pretty much out of time. So yeah, if you guys, it looks like Richard put in the, Richard, is that the sign up?
Yeah, that's the culture survey.
Yeah, Qualtrics survey. And yeah, I'm happy to, if you guys have any questions, I can stay for a little while and wrap up for today. Thank you guys.